Okay, so I'll just hand over to Blanca now, who's going to talk about some analytical. Thank you.
Okay, good morning, everyone. I'm Blanca Espinet. I work in the pathology service in the molecular cytogenetics laboratory in Hospital del Mar in Barcelona. And today I will be covering the, the area regarding the fish probes, types, fish patterns, and basics. And as we have been discussing about this technique, fish technique, I will be covering this part regarding the probes that are the fragments, the DNA fragments that are labeled and are facing the region that we want to study. And first, before starting, I would like to emphasize that we don't have only fish. The labeling of the probe can be different, and in pathology services, for example, in our pathology service, we use also fish. Silver labeling. So fish is the one that I will be covering now, but the probe can be labeled using chromogenic methods or silver enhanced methods. Okay, regarding the types of fish probes, for paraffin sections, for paraffin dealing with paraffin tissues, we have two different types of probes, the Locus specific probes and the Centromeric probes, and I will cover this now. The Locus-specific probes are these smaller probes, or covering a smaller region. In fact, Locus-specific probes cover genomic clones and maybe different in size. The size of the clones, more or less, or the covering regions, more or less, start from 100 kb to more than 1 megabase. And each probe covers a specific gene, specific chromosome locus. Okay, as you can see here, we have an example, a scheme. I don't know if you can see here. This is a diagram in which we can see here the probe covering the TP53 gene. In this case, this probe has 167 kb. And the design of this kind of probes can be from only one probe covering only one region to up to 4, because more or less in fact in commercial probes we can mix up to 4 different colors. Normally we mix red, green, yellow, and aqua blue, so we can combine up to 4 different fluorochromes. And we can combine Locus specific probes with centromeric probes. As you can see here, this one is covering the centromere of the chromosome 17 for the TP53 as a control. And regarding the applications of these probes are mainly dealing with gains losses, so we can see amplifications, gains, or deletions. And if we mix them in a certain way, we can also detect rearrangements such as translocations or inversions. So regarding gains or amplifications, I will show this scheme in all my slides. This is not an error. This is a bit of Spanish or Catalan. Some metaphase, interface, but you can understand. So yeah, this is the first example. It's the control or the normal. So we have 2 copies of each chromosome and we have 2 copies of each locus. So this is a normal case in which we have in red the test, and in the one that we want to analyze, and in green the example is the control. So we have 2 copies each, and in the right part you can see here an amplification in the chromosome and also in the cell, in the nucleus. And the real example, you can see here an example of a glioblastoma with EGFR amplification. You can see here the huge number of the gene. And another example from breast cancer, in which you can see also an amplification, in this case of the HER2 gene. And you can see that the type of amplification is a bit different, because one is more sparse and the other one is more focused, is more local. Here So when we are dealing with amplifications, which is the general definition of amplification, this is a matter of debate, of course, because it's something that we are always discussing about, which is the number. So we have to set the cutoff at 4 or more. In the number of signals that accomplished for an amplification is 4 or more copies of a gene or a gene region, but in fact it depends on the entity, because normally it is associated to prognosis or prediction to response. So it may be variable, what we call amplification. Of course, filtration of the tissue, because if we only have 2 amplified cells in a total of 100 cells in your section, you are not going to consider that case as amplified. And it depends on the tumor. Maybe it has to be at least 10% of tissue, or at least 50%, or at least 20%. So it depends on the tissue. And I've been organizing these diagrams. You can see here 3 copies of your gene and only 2 centromeres or 2 controls. So in this case, it's a gain, but it's not an amplification. Second one is considered an amplification in some cases, as it's like the ratio is 4 copies for 2 controls, so the ratio is 2. But in this, for example, in breast cancer, we have 5 copies and the number of centromeres is 3. It's not considered amplified, as we have 5 copies, but we have 3 chromosomes, 17. So the ratio is not more than 2. Then it's not amplified. And also we can find amplifications like this, like more sparse with many copies, which means that it is probably similar not similar, but I don't know the name in English, responsible or caused by the mean amplification. So that, but in other cases we can find like this one, like clustering. We have clusters that sometimes are uncountable, and in this case we normally say more than 20, more than 100, depending on the size. And it's more similar like a homogeneously staining region. So in these cases, normally the amplification is in the same chromosome altogether, but we cannot see chromosomes here in interface cells. Okay, regarding deletions, as the same as I said before, this is the control scheme. In a deletion, we are losing one of the signals. So in interface, we're going to see this pattern. In this case, I put an example in which we are covering 2. It's not a control. The other one is the test. In fact, we are by both probes. In this case, is the mixture of probes covering ATM and TP53 genes. And in this case, we can see a deletion of TP53, but the other one is not a control. We are also testing the other one. So sometimes the mixture is not control and the test, but are 2 different tested regions. And another example that I wanted to highlight is that sometimes we can find co-deletion or homozygous deletion. So we are looking mesothelioma, in which we are testing CDKN2A gene deletions, and to become positive, we have to find both. So in this case, we are not seeing any signal of our probe. Regarding locus-specific probes, we can mix them to detect rearrangements. The first design that we have, take apart probes or split probes, in which at 5' in one color of a gene and at 3' in another color, just like here. And when it breaks, when this region breaks, it breaks in the middle, more or less. And these 2 probes that are normally together, they become separate. And we can find this pattern, which we call the break apart pattern or split pattern. You can see here an example, a real example of diffuse large B-cell lymphoma, fish being the BCL6 break apart probe with separated signals. However, we can not only detect rearrangements, well, in fact, it is a translocation because BCL6, the rearrangement is a translocation. This type of design can be also used for inversions, as in ALK, in which some of the rearrangements are inversions, like the one that is with AML4, like this one, but we also can find other types of patterns, such as rearrangements plus deletion, in which this example in ALK is also positive, a positive rearrangement, and I guess it will be covered later in the specific entities after the break. And to show you how these genes, these very promiscuous genes, can have many different partners, and this is the this type of design, the break apart, is very useful in such genes like as NIC or BCL6 or Ewing or other, or ALK or ALK or other types of genes that rearrange with many different partners. In contrast, when you have it's always the same partners, the same pair of regions that rearrange together, we have this other type of design, which is called dual color dual fusion design, in which we cover one gene in one color, the other gene in the other color, in different chromosomes, and when they break, they join together and they show this two fusion pattern. And the example here that I have here is also a diffuse large B-cell lymphoma, BCL2 IgH rearrangements. You can see here the 14q region in green, and it is covered with several probes, but covering the whole region. And BCL2, sorry, labeled in red, also all covered. When it breaks in the middle or whatever, here, you will have two different signals, and also in this IgH part, and at the end we will have this pattern. This is the typical or the expected pattern. However, with this type of probes, we can have variant patterns which need to be interpreted, because we will find variant patterns such as this one. For example, in this one of BCL2, labeled in red, 3 copies of the 14q region. This can be an extra chromosome 14 or an extra copy of IgH, which to be an extra 14, but 3 copies, or it could be one of the 14 that is broken and it's splitted, so it may indicate a rearrangement of IgH with another partners. The same we can identify with the red signal. So it can indicate 3 copies of chromosome 18 or 3 copies of BCL2 or one of the BCL2 rearranged with another partner not being IgH, maybe Ig kappa or Ig lambda, for example. We can also find this type of pattern with a BCL2 rearrangement, but with a BCL2 deletion rearranged derivative, or also this one. This is a rearrangement of BCL2 because we can see the 2 fused signals, and probably the other one, the other 14 chromosomes, so the other IgH has also broken and we have split signals, but maybe it's a trisomy 14 or 3 copies of IgH, not a... So this variety of patterns and more have to be interpreted as possible variants, and sometimes will need to be analyzed with other probes or in the context of all the case. Okay. And regarding the centromeric probes, these probes target the alpha satellite sequences that are flanking the centromeres, and these sequences that are shorter are repeated many times, thousands of times in the centromeric region. So we can see larger signals normally because the probe is attached many times to that region. The design again can be alone, so just having the centromeric probe, because we want only to count the number of chromosomes, or more often these type of probes are used as control locus-specific probes targeting for a specific region. And with these probes, we can detect monostomies, trisomies, or other types of aneuploidies. The same diagram, you can see here the normal pattern, the centromere of the chromosome, so 2 copies in metaphase, 2 copies in interphase. In this case, we have 3 copies, so a trisomy. You can find, as I said, some cases, the centromere region, and in this case, a trisomy of chromosome 12 for a CLL. Here it's better because here a lot of signals here. And in other cases, it's very useful to have the centromere because we can distinguish between amplifications and polysomies, for example. And for example, for HER2 or for TP53, no, for HER2, this is wrong. I wanted to put here the HER2, and it's an error. In fact, for HER2, this is a HER2. You can see here like 5 copies of the HER2 gene, but more than 5 or 6 centromeres. So this is a polysomy case. And then we can combine multiple probes, as in this example, the melanoma test, in which we are combining 5 locus-specific regions, targeting 6P, 6Q, and 11Q, cyclin D1, and the centromeric region for chromosome 6, and we can find results like this. Of course, we are not analyzing all together. We are analyzing fluorochrome by fluorochrome, but at the end, we have to deal with all this information together. Okay, and finally, to close, I would like to say some words regarding how we count signals. As we have heard before, it's very important, it's essential to select the tumor area because we want to know where are our tumor cells. When we deal with a suspension, we don't mind if about the selection of an area because all the cells are homogeneously in a suspension, but it's not happening. So we will need to go to the slides and have the tumor area previously marked using hematoxylin slide or immunohistochemistry slide. Or some cases we can go to the screen and like match your slide and mark the region in order to be able to test on this specific region. It's important to, once it has been hybridized, to screen the whole slide to check fish performance in general, to be able to say, okay, this is a good slide, I can count, I can analyze this slide. It's very important to identify normal, which I mean control for us, cells in tissue. In this case, it's a SISH, silver enhanced hybridization. And it's a HER2 case with an amplification here. These are the tumor cells, and marked here we have the fibroblasts, which are the normal cells, and we can see the signals there. So we are sure that these cells have hybridized correctly, and this is how we can say, okay, the hybridization is okay, and we trust in these signals because we see the normal cells, and the normal cells are good. Well hybridized. Okay, We also have to take into account the shape of the cells, the type of cells, the shape of the cells, if they are rounded or fusiform, depending on what we are looking for. If we are, for example, this is an example of fish in a dermatofibrosarcoma protuberans in which the cells are fusiform, like this type of shapes. So we are going to find these areas. But for example, in lymphomas, we are looking for the rounded cells. So it's important to know more or less which is the morphology that we are looking for or what are we expecting. And regarding the size of the cells, it is also important, and you have to be in touch with your pathologist. For example, in lymphoma, sometimes we have small cells and big cells, and the large cells are the ones that are tumor, but in some other cases, maybe you have to go to another region, and in this region you have like a mixture between large B-cell lymphoma and follicular lymphoma, and you will need to analyze or to score both regions. So it's important to know before what you're looking for and which is the size and the shape of the cells that you are going to screen. And regarding the type of the it is important also to know which is the shape that we expect of the signals, because sometimes we can find good spots very defined, but in some other cases we can see like this type of more sparse or like different, not very well defined spots. So we need to know how is the hybridization, if we trust on this hybridization or not. Of course, we need to know which are the patterns. So we expect to have two fusion, one red, one green, but we are seeing another different pattern like the ones with 2 red, 3 green. So what's this indicating us and how to take this into account? And again, we will need to deal with the distance between signals. And of course, important to check the probe designs, because sometimes this distance between probe signals, maybe it is said that more or less we will take in mind that it can be a distance between them and it's not separated, it's normal. But of course, sometimes some designs can be difficult. And I wanted to show here the MIC break apart designs, which are very variable because the region the gene can be broken is very wide. So in some cases, like the Abbott probe, the distance is the base between the two red and green signals. You can see here the MIC gene. Beta system is smaller, and with DACO, smaller. And here another design with 3 colors. So you have to take in mind which is your design and if the size in between is wide or not, because sometimes you can find cases like this in which you may say, okay, this is a rearrangement because we see a space here in between. But I have to say that this case was normal, was not rearranged. And we could see this type of signals, separated signals, but this was not rearranged. And of course, you can see this white separation between signals, and this case was positive. So this was a positive cell, and this, which we can see also a separated signal in between, was negative. Or in this other one also was negative. So it's important to know which is the design of your probe, able to deal with these situations. To finish, important to know that we are losing signals due to sectioning. So the tumoral size, the median size of the diameter of a tumoral cell nuclei, and this is general, because you know that tumoral cells can be bigger or larger or smaller, but it's more or less 6 micrometers. But when we are dealing with tissue sections, the tissue sections are between 3 and 4. So we are going to find like this high number of loss of signals. So maybe we can say that we have lots of deletions, but these are not true. So the cutoff for deletions will be high. And to trust on a deletion, we will need to find at least 30 or 40% of cells with this deletion in our case. If not, we are not sure. And of course, we will see heterogeneity on the copy number due to sectioning. So maybe it's not happening in all cases, but we face this a lot in lymphoma because the cells are more like together, and it's more difficult because the tissue is like very tight. And sometimes it's difficult to deal with deletions in paraffin sections. Finally, the total number of scored cells. It depends again on the tumor type. In general, we score at least 100 cells, but depending on the type of tissue, in general, we report the pattern, but with the expected pattern, we say we do like similar what the pathologists do, like more in general, like say 80% or 50% or 60% of our rearrangement, because we screen all the slide or the region. But for example, for deletions, we normally count. In breast cancer guidelines, the ASCO-CAP guidelines say that you should count at minimum of 20 cells and calculate the ratio between HER2 and centromeric probe of chromosome 17. In melanoma, the minimum is the number of cells is 30, and the complex analysis criteria are there. So we need to analyze different aspects. Some of them are ratios, some of them are percentages of amplification or gains or whatever. In lung, minimum of 50 cells. In brain, minimum 100 cells. So it depends on the guidelines. So I think that we should follow the guidelines for each specific tumor, and it is the most important thing that I wanted to say in this slide. And finally, I just wanted to acknowledge people from my lab, because here we have the technicians and they are working a lot and working hard with the pathology service, with the paraffin sections, and also Sergi Clavé, who is now in the molecular diagnostics labs, who did a lot of FISH also on lung cancer.
So we'll combine the question section for this presentation with Sharon's at the end.